This is part 2 of my series on catalysts, talking about enzyme catalysts. Now, human physiological processes have to be maintained at a very narrow temperature range. If the temperature is too low, below 300 Kelvin, the reaction is too slow, and that leads, of course, to death. If the temperature is too high, the molecules begin to decompose, and that, of course, also leads to death. Enzymes and coenzymes developed or were created in order to facilitate the biological reactions. And because of hydrogen bonding, breaking, if you get much higher than 300 Kelvin, then those hydrogen bonds break. Now, remind you of how these enzymes form. Peptides, amino acids, polymerize, and they polymerize into polypeptides. These long chains, fold into proteins. The proteins then fold, and a lot of the time other proteins come in to add to the structure, and they make enzymes, and these enzymes then catalyze a narrow range of reactions. Each enzyme is going to have usually one active site for catalysis. The active site is a pretty small section of the molecule. It may involve peptides that are hundreds of units away from each other, but because of the folding, they bring these peptides close together. Generally, the folding is done through hydrogen bonding, which lowers the overall energy of the combination. Then the substrate molecules, as you learned in first-year biology, are going to fit into the active site like a key in the lock. Here is a model of a substrate molecule, in this case the two molecules ADP and the pyruvate with phosphate, fitting into an active site. And as you can see, there's a kind of a pinching effect by the enzyme, which forces the phosphate here to attach to the ADP and make ATP. And then the pyruvate, which is the non-hydrogen part of the pyruvic acid, is kicked out along with the ATP. Now, coenzymes are molecules that work with enzymes to make them active. All vitamins are coenzymes. For example, vitamin B6 works with D-amino acid aminotransferase, the enzyme. This is a model of the atoms put together into that complex. Here you can see the coenzyme fitting within the active site and actually making the active site active. How do enzymes speed up a chemical reaction? Well, as you saw with the inorganic catalysts, they provide a site, a place for breaking bonds and then making new bonds. The enzyme typically attracts the substrate and steers it using electrostatics. The electrostatics help to explain why amino acids are uniquely qualified to make up enzyme molecules, because amino acids are all polar. They have the acid end and the amine end, and many of them are zwitterions. They have a plus charge on one end and a minus charge on the other. Enzymes themselves undergo conformational changes as they bond with the substrates. The changes improve the fit, but they usually raise the energy of the enzyme-substrate complex, making it more reactive. The enzyme-substrate interaction requires spatial complementarity and chemical complementarity, or compatibility. So, for instance, complementary charges on nearby groups, so that plus attracts minus, or complementary polarities on nearby groups, so that the minus end of one group is attracted to the positive end of the other, or nonpolar regions on nearby groups that will help to steer the interaction because they are not particularly attractive to anything that is positively or negatively charged or has a positive or negative polarity. And then, of course, hydrophobic or hydrophilic compatibility, and that has a lot to do with polar versus nonpolar. Here is an example of a simulation where an enzyme induces a charged region on the substrate. So here's your substrate, here's your enzyme, and as they come together, the substrate acquires a partial charge on one of its regions, perhaps by induction. Enzymes will form numerous and precise weak bonds, mostly hydrogen bonds, with the substrate. So here is the enzyme surrounding a substrate. And you can see these various bonds forming, involving usually polar ends of the amino acids, like this NH3 plus on a lysine here, and another one there. Here is a blow-up of the previous slide so that you can see the interactions that are going on. For instance, this magnesium 2 plus with the negative end of these hydrogen bonded, or excuse me, these OH group. This is a CO2 group from a carboxylic acid. Enzymes also lead to, when they form with a substrate, they lead to a transition state. The transition state features the tightest bonding, but it also has the highest energy, and therefore it is the least stable. And that then causes the enzyme substrate to break apart, and the product then is kicked out from the enzyme substrate interaction. This brings on the breakup. Here is a good example of covalent interactions, and you can see this enzyme and this substrate. Here's a blow-up of various parts on serine and histidine, the amino acids within the protein. Here the substrate, this carbonyl carbon, is being attracted to this part of the CH2OH on the serine. A bond breaks and begins to form here, so that this bond is actually coming over to make a bond here. You can see now the bond to the carbon and oxygen, the new bond is formed to the enzyme. It's a covalent bond, a sigma bond, and hydrogen now has been kicked out, and here is one of the products of the reaction. Water molecule is attracted to, this is the hydrophilic part, is attracted to the C double bond O, which has a slightly positive charge right here, and the negative part of the water is attracted to it, and a bond is formed. The hydrogen from the water is left on the enzyme, and as you can see, there is no overall change to the enzyme, but the hydrolysis has been completed. Enzyme activity is very sensitive to changes in temperature. Heat disrupts the weak bonds by increasing the energy of the molecular orbitals, and heat changes the conformation of the enzyme coenzyme, usually by the breaking of hydrogen bonds. Heat can disrupt the active site and reduce the reaction rate dramatically. That's why you do not like fevers to be much over 102 Fahrenheit for any length of time. As you can see, the enzyme activity against the reaction temperature in Celsius, and when it gets into this region here, it's highest, and then it rapidly falls off and is essentially dead at 100 degrees Celsius. Acidity changes also affect the charge balance. It affects the polarity of molecules. So pH changes can really rack up an enzyme interaction. It disrupts the active site and reduces the reaction rate. Enzymes appear to be designed for a narrow range of pH. They will not function outside that pH range. This gives you an idea for 2 of these, what the pH versus activity is. Now, if you introduce a competing substance, that can also interfere with the enzyme-substrate interaction. You may recall that carbon monoxide interferes with the oxygen-hemoglobin complex and actually will bond better to hemoglobin than oxygen. Competitive inhibition is the introduction of a valueless substance that's similar to the useful substrate, and it essentially stops the action of the enzyme and can cause the cells to even die. It blocks the active site, especially if the fit is better than the enzyme substrate fit. There's something called non-competitive inhibition. All inhibitors prevent binding of the substrate to the active site, but non-competitive inhibition introduces a molecule which binds at a nearby active site and prevents the useful substrate from binding at the proper active site. In other words, it gets in the way of the enzyme substrate forming. Or ultimately, it could bend the enzyme so that the enzyme cannot do its job. To end, I'd like to show this ATP synthase. You'll notice up here that a high hydrogen ion concentration is pushing this engine, this ATP synthase engine. You can see the hydrogen ions coming down here and going into this rotor. The rotation of the rotor causes this cam, this is a part of a protein, to knock these other protein subunits. Phosphate is coming into the active sites here, and ATP is being kicked out. ADP is coming into the active site here, and along with a phosphate, as you can see, energy is added and it's kicked out. This rotor is actually acting kind of like a cam on an engine to make the process go. In your cell, you can see that these rotors, thousands and thousands of them in the mitochondria, are actually driving the overall process and the energy of your cell.